Hello, my name is CPRoussado and I'm doing my PhD in the Center for Stress and Age-Related Disease at the University of Brighton. And I would like to share our work entitled Nanopore Sequencing Approaches to Profile the Gut Microbiota of Mice Infants Exposed to Alcohol in Utero. The presentation is divided into 5 main sections where I will guide you through the work we have done. The consequences of prenatal alcohol exposure represent a major public health issue, having a wide-ranging effects at a staggering economic cost. Fetal alcohol spectrum disorders (FASD) represent a cluster of abnormalities, including growth deficiencies and neurologic impairments, which are not easily diagnosed. In the picture, we can see the most severe of FAST conditions, termed FAST, fetal alcohol syndrome. This is why it is important to increase the knowledge of what happens during gestation. The entire perinatal period is a critical development window characterized by rapid evolution of gut microbial colonization alongside changes in neuronal organization. One of the factors that influences this complex interaction is mother's diet. Alcohol can trigger gut dysbiosis and promote many malfunctions, including liver disease and disruption of the gut barrier. The figure shows the summarized impact of gut dysbiosis in alcohol-related liver disease. Nanopore sequencing is a third-generation sequencing technology that works by crossing DNA molecules through nanopores embedded in a membrane. While crossing, the membrane's potential is shifted, and those changes can be measured, indicating which base was crossing at each given moment. Albeit the rising in the number of publications observed in the recent years, challenges remain. For instance, high error rates, around 10% in some cases, and non-standardized analysis tools. The objective of this study was to assess the effectiveness of target and untargeted nanopore sequencing approaches to profile the gut microbiota of infant mice exposed to ethanol in utero. Since the clearest data regarding the role of the gut microbiota on neurodevelopment and psychiatric disorders came from mice, we relied on this model, therefore allowing insights that were not possible to study directly in humans. For the methods and workflow, DNA was extracted from the guts of 12 mouse infants born from mothers that received alcohol in their diet in a way that mimics average human alcohol consumption. For our study, we used the MinION device for sequencing 3 different libraries during a 24 hours sequencing run. 2 targeted libraries and 1 untargeted metagenomic library were generated with the ligation sequencing kit, and the 12 samples were barcoded accordingly. For the targeted approaches, amplicons were generated through PCR amplification of the full-length 16S rRNA gene with 1,500 base pairs, and the 16S 23S ITS region of the entire rRNA operon with 4,500 base pairs. The sequencing data were then base called and demultiplexed with Guppy, classified with What's in My Pot, and further analyzed with Microbiome Analyst. For the results, in the targeted approaches, the average read length was similar to the size of the respective amplicons, which is a good indicator that nanopores were indeed sequencing the desired targets. In the metagenomic approach, average read length shows that DNA was fragmented, avoiding the sequencing of longer fragments. Based Based on the quality scores, we can see that in the targeted approaches, error rate was between 8.3 and 9.7%, and in the metagenomic approach between 8.1 and 8.6%. Additionally, the metagenomic approach had a higher number of unclassified reads. In terms of microbial composition, the three approaches yielded different profiles. However, we observed that the same two species were the most abundant in the three approaches, Duncaniella spp8 and Muribaculum intestinale. On the left figure, we can see that the three approaches yielded significantly different taxa diversity. The 16S approach yielded the most diverse gut microbial composition, and the 16S 23S ITS approach yielded the least. On the right figure, we can observe that there are a clear microbial taxa clusterization within samples, here, here, and here. This means that the taxa found in each approach are similar and related to the approach chosen. Moreover, this approach taxa association is also observed at the taxonomical level, as we can see exemplified in these two figures. From blue to red means an increased positive correlation. Pearson correlation coefficient was used here. On the left, we can see that actinobacteria positively correlates with the metagenomic approach, and Firmicutes negatively correlates with the same approach. On the right, we observe that bacteroids Caesimuris, Curtobacterium flaccum and Bifidobacterium animalis who positively correlates with the metagenomic approach. Additionally, Lactobacillus johnsoni, Limosilactobacillus reuteri, Amedibacterium intestinale, and Peptinophilus harei negatively correlates with the same approach. Other slighter correlations were also observed. Therefore, we may conclude that choosing a nanopore sequencing approach might over and underrepresent specific taxa. However, this observation needs to be further investigated. In conclusion, differences or biases might arise depending on the methodological approach chosen to perform nanopore sequencing studies, then confounding the real microbial profile of original samples. Next, optimized nanopore sequencing approaches will be used to assess the disruption of the mice gut microbiota after exposure to alcohol in utero, contributing to advance our knowledge on fetal alcohol spectrum disorders. Thank you. Finally, I would like to thank all the authors of this study, University of Brighton and European Union's H2020 Research and Innovative Program for the funding, and the organizers of this Congress for the opportunity to share the results. Thank you.